The next topic of my presentation is phenotypic characterization of compound effects on liver spheroids using confocal imaging and 3D image analysis. This work was done in collaboration with Cellular Dynamics International. This work was done using iCell hepatocytes from Cellular Dynamics, and those are highly pure population of human hepatocytes derived from induced pluripotent stem cells. Those cells have typical liver morphology and also express a number of important markers, including albumin, occludin, and number of others. iCell hepatocytes enable access to commercial quantities and also high quality of cells, which are useful for preclinical drug discovery, hepatotoxicity, and disease modeling. It is believed that 3D cultures more closely resemble in vivo conditions because cells normally grow and interact in 3D. It may also better mimic microenvironments and provide different cell morphology and function. In addition, spheroids allow assay miniaturization and use less experiment. From the other side, 3D cultures require different assay workflow and different methods for detection and analysis. Studies done by Cellular Dynamics demonstrated that for certain compounds, 3D cultures could be more sensitive than 2D culture for assessment toxicity. The goal of present study was to develop and evaluate fast, accurate, and reproducible high-content imaging methods which would allow to investigate effects of toxic compounds on morphology and viability of 3D liver spheroids. The workflow of the assay presented here. Human iCell hepatocytes were cultured for 7 days in 2D format, then detached and supplemented with extracellular matrix and plated into ultra-low attachment plates, either from Corning or in Spheroid. Both plates eliminate spheroid transfer steps and center spheroids in a small focus area, facilitating capture of entire spheroid in one 10x image. Cell aggregated at the bottom of the well, forming spheroids in 24-48 hours. On this slide, you can see very significant differences in metabolite secretion, whether it's in 2D or 3D culture. You can see much higher level of secretion of urea and also other metabolic enzymes. Spheroids were stained, as we described before, using mix of 3 dyes, Hertz, calcium AM, and ethidium homodimer. Images were acquired using 10x or 20x objectives, so 10 to 13 images were obtained 5 to 10 microns apart. In addition to individual images, maximum projection was also saved. Spheroids were treated for 72 hours with compounds which cause liver toxicity. In this example, you can see control spheroid and also spheroids treated with taurosporin or ketoconazole or haloperidol. You can see marked phenotypic changes caused by compound treatment. For example, you can see decrease of live cells, calcium positive, and increase of the content of ethidium homodimer positive cells, dead cells. You can also evaluate apoptosis or mitochondria integrity using the appropriate dyes. Spheroids treated with compound increase number of apoptotic cells, which positive for caspase C7. And in other example, you can see decrease of mitotracer orange, which stains cells with intact mitochondria. 3D analysis of images was done using recently added features of MetaXpress software. This software converts a stack of 2D images obtained by confocal acquisition into 3D space with appropriate detection and segmentation of objects. Spheroids can be characterized by their size and also intensities. For example, spheroids treated with methylmercury are very different from control spheroids by their diameter, volume, and also intensity of FITC stain. Software also allows to detect small objects within spheroids. Here you can see example of count nuclei segmentation and cell scoring analysis. Custom model editor usually used for analysis, which allow to design number of steps, allowing to detect and quantitate and characterize nuclei, cytoplasm, and cells. Individual nuclei segmented in different planes, then combined in 3D space using connect by best match function. Cytoplasm, mitochondria, or other features could be also segmented and defined in 3D space. Significant changes of spheroid phenotypes is observed after compound treatment. So you can see changing of shape, spheroids, their volume, and also content of different cells, either live or dead. So those phenotypic changes could be quantitated and measured by software. Quantitative analysis of images included derivation of parameters to assess morphological features of spheroids, cell content, and complexity. The volume, diameter, intensities were measured, and also the number of calcineurin positive cells, live, or number of ethidium homodimer positive cells, dead, and also average volumes and intensity of cells showing these markers. 3D analysis allows converting information about cell segmentation into 3D space. Nuclei or individual cells could be counted or characterized within larger objects, in this case spheroid. It is also possible to characterize and score cells for live and dead using calcineurin staining, and also define not only number but also volume and volume sum of cytoplasm of cells positive for calcineurin. You can measure different kinds of phenotypic changes, including decrease in a number of live cells, decrease in a volume of live cells, and also intensity of calcineurin staining. Spheroid diameter or volume or number of dead cells has significantly changed as a result of compound treatment, but usually did not follow for parametric concentration response. In contrast, the number of live cells as well as the volume of live cells or calcineurin intensity had a great assay window and also good fit into 4 parametric concentration response curves. Spheroid assay performance was characterized using a representative set of 48 compounds that included some known cytotoxic and hepatotoxic agents and also 6 chemicals considered safe or non-toxic. The table represents IC50s for different classes of hepatotoxic compounds, which include known liver toxins, anti-cancer drugs, antidepressants, and several other types of drugs. We compared IC50s for 3D cultures and 2D cultures. You can see some notable differences in IC50s for several compounds when other IC50 values are not significantly different. We need to reiterate that exposure time was 72 hours, which is acute exposure, and there is a possibility that longer term of culturing may cause more differences. We also compared effects of 23 selected compounds on spheroids made from transformed liver cells. There were dramatic differences between datasets, especially for anti-proliferative compounds. The IC50s determined for Hep-G2 spheroid cultures were in nanomolar range, when for iCell hepatocyte spheroids, IC50s were much higher. In contrast, the majority of other hepatotoxic compounds showed similar effects in both cell models. So the rank order for toxicity was very different because Hep-G2 spheroid cultures ranked the anti-proliferative compounds as more toxic. In the next study, we have developed methods for formation of 3D cardiac spheroids derived from human-induced pluripotent stem cells. Cryopreserved iCell cardiomyocytes, 2 cells from cellular dynamics, were used in the study. Cells were plated into U-bottom 96 or 384 well plates from corneum, and cardiac spheroids were efficiently formed from those cells, typically within 48 hours, and they started contracting spontaneously. These 3D models could then be used for cardiotoxicity studies by staining them with calcium sensitive dye. Changes in fluorescent intensity in response to intracellular calcium flux were used as surrogate marker for spheroid contraction. Significant alteration for the baseline beating pattern was observed in response to cardioactive and cardiotoxic compounds. Spheroids were loaded with calcium-sensitive dye and treated with different compounds. In this example, you can see control spheroids and also spheroids treated with isoproterenol or cisapride. There are a number of parameters which changed by compounds, which include changing in a beating rate and amplitude, changes in rise or decay time, and also peak spacing. Different calcium flux patterns can be recorded by imaging instruments or by flipper instruments. The pattern can be analyzed by software. Different parameters could be used to measure concentration dependencies and define IC50s for different compounds. In this picture, you can see those dependencies for beating rate for several benchmark compounds.